acid. So that allows for some flexibility in that third base pairing position there. Okay, and so that third base pairing position there is called the wobble position. For instance, a U on an anticodon, okay, can bind with an A or a G on a codon. Okay, and so that's that wobble position, which shouldn't change the amino acid that should be there. So we're going to have to get these amino acids actually attached to the transfer RNAs. And they're going to attach to the transfer RNA using what's called a charging enzyme. Okay, and so the charging enzyme in this case is our amino acyl tRNA synthetase. Okay, and so that's going to be what's called, like I said, our charging enzyme. And this is going to actually put the correct amino acid on the transfer RNA based on the anticodon. So here's my amino acid. Okay, my amino acid, and you notice this process requires ATP. So this process requires energy. So my amino acid gets phosphorylated, okay, in the charging enzyme. The tRNA is then able to come over, and the enzyme will facilitate the attachment of the tRNA to that amino acid. And like I said, that's going to require energy. That's going to require ATP. Okay, but it's going to produce what's called an activated amino acid. And then that amino acyl tRNA synthetase can be reused. Okay, just like transcription, translation has an initiation phase, an elongation portion, as well as a termination portion. Okay, so first we have to initiate, we have to start the process. Okay, and so initiation is going to start with the small subunit of the ribosome attaching to the messenger RNA at the start codon. Remember, the start codon is going to be AUG. It's going to be that it's going to code for methionine. Okay, and so the small unit's going to attach, subunit of the RNA is going to attach to the messenger RNA at the start codon. Okay, that's also going to bring that transfer RNA in, okay, that will have the UAC anticodon on it with the methionine to it. That's going to attach at the P site. Remember, the P site is the one in the middle, okay, because now the large subunit is going to have attached. So when the initiation is done, when the initiation complex is formed, we've attached the small subunit, the large subunit, and there is a tRNA in that middle P site, so that there's room for the next tRNA to come into the A site to attach. So once we've started this process, now we have to elongate the polypeptide chain. Okay, so the transfer RNA will enter at the A site. So the next tRNA comes in there at the A site, and they will form a peptide bond between the amino acids. So the codon recognition will occur, okay, and that's going to require an input of energy. So there's an input of energy in this elongation process. Okay, and now that you have a tRNA at both the A and the P site, the peptide bond is formed. Remember, the peptide bond is formed between the amino acids. Okay, so that's my peptide bond between the two amino acids. These little lines here, these are all peptide bonds. Okay, and so the peptide bond will form between the amino acid. That's going to actually release water when we form the peptide bond. Okay, it's a dehydration synthesis there. So I'm releasing water, and now everything will shift down. Okay, again, it requires an input of energy for this what's called translocation to happen. Okay, and all of the tRNAs will shift down. The tRNA that was in the P site now no longer has an amino acid attached to it. So it'll shift over to the E site where it can be ejected or exit from the ribosome. The tRNA that was originally at the A site, okay, so it came from here, is now going to move over to the P site. And now that A site is free for the next tRNA to come in and bind to the codon that is next. And that also allows this transfer RNA that was released from the E site to be able to go pick up another amino acid. Transfer RNAs are reused. They are not consumed. So the elongation process will occur until what's called a stop codon is reached. Okay, and so a stop codon is going to code for what's called a stop protein, okay, or a release factor. Okay, and so that stop codon, that stop protein, that release factor, is going to attach to the A site. Okay, you can see it here blocking the A site, so nothing else will be allowed to come in there to that A site. That causes the very last tRNA to be released, and you'll notice it's let go of the peptide chain that it was holding on to. Okay, the polypeptide chain now separates. The ribosome separates into the large and the small subunits. Essentially, everything breaks apart. Okay, and so translation has now effectively stopped. And now we have this polypeptide chain that if it needs to be processed farther, it can be processed farther. If it needs to be used right there in the cytoplasm, it can be used right there in the cytoplasm. So some proteins get exported from the cell. Okay, so they are, for instance, those protein hormones, okay, that are going to go somewhere else to do their function. So some proteins need to be exported from the cell. And the proteins that need to be exported from the cell, they are tagged and interact with these signal recognition particles, these SRPs. And so what will happen is these signal recognition particles, they are going to attach on to the growing polypeptide chain. So you can see translation is occurring here. The messenger RNA is threading into the ribosome. You can see the polypeptide chain elongating, and they will help guide that polypeptide chain into the endoplasmic reticulum. Okay, so this is my inside. Remember, our endoplasmic reticulum was like a network of tubes. Okay, so this is the lumen. Okay, or the inside, the hollow space in the endoplasmic reticulum, and so they will help guide that growing protein polypeptide chain into the lumen. And one of the jobs of the endoplasmic reticulum is to continue to process proteins that are going to be exported. Remember, they'll get modified, then they'll get shipped over the Golgi apparatus and get modified and packaged for shipping out. Okay, and so these signal recognition particles are what help get it into the endoplasmic reticulum so that process can happen. So let's take a minute and look at some of the things that could go wrong, some possible mutations that could happen here. Okay, so first of all, let's look solely at what are called base pair mutations. So something is going wrong essentially with just one base pair. Okay, so we can have a mutation that causes no effect at all. Here's my original up here. This is my amino acid chain. Okay, I've got a mutation right there. I've got a substitution. But because it's in that wobble position, it's at that third base position, there's a chance that it doesn't do anything. And that's what happens here. That still codes for glycine. And so no one even ever know this mutation existed. Okay, sometimes those are called silent mutations. Okay, because they don't do anything, so you don't know that they're there. Okay, you can also have what's called a missense mutation. Okay, so a missense mutation is going to change one amino acid. Okay, so out of the list of amino acids, it just changes one of the amino acids, which can be a huge issue. Okay, and then you have also the third kind is what's called a nonsense mutation. Okay, a nonsense mutation is going to shorten the polypeptide chain. It's going to code for a stop codon, and that's it. It stops. Nothing keeps going after that. It stopped. It signaled the attachment of that release factor, and so the subunits have broken apart, the polypeptide chain has been released. Okay, it stops the formation of the polypeptide chain. So these can have a very large impact, okay, because a lot of times they will make the polypeptide chain significantly shorter than it used to be. Let's look at a couple of examples. For instance, cystic fibrosis. Okay, cystic fibrosis is an example of, we've had a deletion of 3 nucleotides. And so by deleting those 3 nucleotides, that causes the loss of an amino acid. So there's this big, long polypeptide chain. The 508th amino acid of that polypeptide chain is missing, and that results in cystic fibrosis. Another disease example would be sickle cell anemia. Okay, so sickle cell anemia is a point mutation. So there is one amino acid gets changed. Okay, it's a missense mutation. There is, on the hemoglobin protein, a glutamic acid is substituted for a valine. So there is a change in the amino acid sequence. There's a change in one amino acid. Okay, a glutamic acid is substituted out and a valine is in its place. which makes the hemoglobin chain a little slightly less soluble in the cytosol, okay, which has an impact on the shape of the hemoglobin chain and so oxygen delivery for the organism. Other kind of mutations are what are called frameshift mutations. Okay, these are going to be due to an insertion or a deletion of nucleotides. Okay, so in this instance, you could have a missense, which just changes one nucleotide. Okay, so you have you change one amino acid, so you insert or delete it, and it manages to change only one amino acid. That would be very rare in these particular types of mutations, okay? Because as a general rule, with a insertion or deletion, you're going to you shift what is called your reading frame. Basically, I shift my grouping, right? Here, these are grouped in threes like this. I add one in, and everything after it now changes how my threes are grouped. Okay, so these are going to be a result of an insertion or a deletion of these. You can have a large insertion or deletion. In this instance, you've got three nucleotides that are missing. So this isn't a frame shift. I didn't change every single thing that came after it. I still get phenylalanine, glycine, okay, but I am missing an entire amino acid, which could be a problem. And you can also, again, have a frame shift that causes an immediate stopping, a nonsense mutation again. Some more examples here would be Tay-Sachs. Okay, so Tay-Sachs is a disorder where there is lipid or fat accumulation in the brain, okay, and that is usually broken down by an enzyme called hexosaminidase, and that enzyme, okay, is coded for. And what happens is there's a 4-base insertion on the hexa gene, and so that causes a nonsense mutation. And so it makes one of the subunits of the hexosaminidase essentially deficient, and that causes so that it's unable to break down those lipids, causes the buildup of lipids in the brain. Tay-Sachs is a lethal disease because of this small mutation. But one of the things we need to keep in mind is that not all mutations are bad. Some mutations can be beneficial. They lead to new genes. They lead to new proteins. That's how evolution and adaptation occur, okay, is with mutations. New genes, new proteins altering what is currently there. So we'll be working with some models for protein synthesis in class.